Okay, so hello, I am Elizabeth Boulding, and this is Newcomb's periwinkle, Litorina subretundata. And what we're going to investigate today is whether this temperate, poorly dispersing periwinkle is likely to evolve the ability to survive extremely high rock temperatures. And this is interesting because you often hear in the news about adaptation and sort of climate adaptation as if everything's going to be able to adapt, which I find quite irritating because I don't believe it's true. So here we are. Here is Vancouver, Victoria, Barclay Sound. Here is Banfield Marine Science Center and my two study sites on the Hawaii First Nations land, Praiseola Point and Nudibranch Point. And this is the Pachina Campground. And this is a site that's very highly wave exposed, which makes the ecology much simpler. And just as an aside, even though I'm not going to talk about it today, the reason that I'm monitoring these sites is I have a long-term predator addition experiment that's been quite successful. So because of that, twice a year, we go to this site in August and in December, and we have permanent quad rats, and we count the quad rats, and we put all the snails bigger than 1 millimeter into dishes, take them back to the lab, identify them as species under the microscope, measure them, and count them, and then bring them back and put them back. And here are two of my field assistants, my niece, Paige Stewart, and Caitlin Burry during the COVID lockdown. And here are three of Newcomb's periwinkle hiding among the barnacles. So at these sites, we're finding 4 species of snails. 2 of them are poor dispersers, and the larger Litorina siccana is not very common in this area. But there also are 2 pointy ones that have not just a floating egg mass, but also have a larval form, whereas these 2 do not have a larval form. So if we lump the 2 planktonic species together, what we can see is that the times when their recruitment, so this is the 2 and 3 millimeter snails are most abundant, is right after a very strong El Nino event. So we see this in 97, 98, and again in 2015, 2016, and these are the estimated marginal means. But we almost see an inverse relationship. We see that when the direct developer, Newcomb's periwinkle, is common, generally the recruitment is rather poor for the planktonic developers. And in another paper, I've investigated some hypotheses and managed to eliminate some of these. And I don't have time to go into that because I started a bit late. But what I'm going to talk about today is hypothesis 3, that first of all, the reason that the direct developer is not common is because the warmer air temperatures, it has egg masses on the rocks, unlike the pointy ones, and therefore they're quite likely to get baked in the warmer air temperatures and lower waves that happen with a very strong El Nino. And also, it's not just the juveniles. What I noticed after the 2021 heat wave event, which is really extreme, was that the adults were dying too. So this shows we've had loggers since I had a postdoc in 2000, and she got us hobologgers, and I've been using them ever since. So we know in 2005, the hottest temperature that these loggers recorded was in 35 degrees, and that's quite different from what we recorded during the heat event in 2021. For the first time, we got temperatures well over 50 degrees, and we got them with both replicate loggers. And you can see that a poor snail who was next to this logger would experience temperatures well above its heat coma temperature of 35. It would have 40 degrees for 75 minutes, 45 degrees for 75 minutes, and 50 degrees for 45 minutes, which is very likely a lethal temperature. And we see that it's even worse at the other replicate logger. Now, what stops this rock from getting even hotter? Well, eventually the tide comes in. But as you see, it doesn't completely, it comes in, but it's not just the tide. What happens is there's also some surf. And what the surf does is the surf brings down the temperature to that of the ocean fairly quickly, which is somewhere between 10 and 15 degrees. But nevertheless, what we're seeing is most years, these snails do not experience temperatures of over 45 degrees. We did get a few that were above 45 degrees, especially in 2010. This is the number of minutes per day, which is probably what's relevant as far as killing them. But in 2021, we got a lot of these, and they weren't just 45 degrees. They were well over 45 degrees. And what we can see here is in 2021, the average temperature, when you only looked at those above 45 degrees, was nearly between 47 and 48. So the question is, if you look at this snail, and this is the heat coma temperature estimate that my PhD student did sort of as a side project, it's around 35 degrees. And it has a standard deviation, which you can calculate from this, of about 1. Now, compare that to tropical relatives, which are shown here, and they have heat coma temperatures nearly 10 degrees more, 45 degrees Celsius. And usually the lethal temperature is about 5 or 10 degrees above that, which is shown in the squares here for some of the species. So the question is Newcomb's periwinkle, which after all, it can locally adapt because it doesn't have very high dispersal rates, is it going to increase its heat coma temperature by 10 degrees? Well, even the tropical snails have problems, and I'm not going to go into this in great deal, but they're also experiencing above their heat coma temperature. This one, in a short study, they found temperatures of 48, which is above 45, and 50. So clearly their behavior, like being in cracks, pointing their apex towards the sun, and various tricks they use is really important. But the other thing that's important is having the right enzymes. And we know that active site amino acids are very, very conserved. But sometimes you can have another site, an allosteric site, and apparently the amino acids at those sites can evolve faster because they're less conserved. So if it behaves this way, it would seem probably not. What happens is when an enzyme gets too hot in an ectotherm that's evolved to perform optimally at a much lower temperature, is this active site no longer functions. Now, the other approach we can say is, well, we can't figure out what all those enzymes are doing anyway, so we can treat it as a quantitative trait, a single quantitative trait, and think more about the behavioral ways that they might be able to avoid temperatures of 45 degrees. And this is a model we did a long time ago, my husband and I, and basically what happens when things get really bad in a small population and they become maladapted is most of the individuals die. But as long as some of those individuals survive, and if the ones that survive are the ones that had, because of the standard deviation, a slightly higher heat coma temperature, what you would expect is over time, these ones that are more resistant would breed with one another, and the population would recover and this is basically the 35 degrees, is you subtracted 35 degrees, so basically you would evolve to the new heat coma temperature. The question is, it's not clear whether this kind of model, which predicts could evolve as much as 12 degrees, is really applicable in this case because it's not clear the enzymes really obey the assumptions of quantitative traits, which is many loci of small effects. So it doesn't look very good. And what seems more likely is one of these species that is a good disperser that's already adapted to warmer temperatures will invade, and this species will go extinct. So just my conclusions so far, rock temperatures of over 45 degrees have only occurred in 7 out of 20 years of logger data. And temperatures of over 50 degrees, which is well above the heat coma temperature, it only occurred in 2021. The maximum rock temperature often exceeded the heat coma temperature of 35 degrees plus or minus about 1 degree. That's the standard deviation. And these snails also show some behavioral modifications. They hide in cracks, they hide under barnacles, they hide in the seaweed. But if it gets up to 50 degrees, it doesn't look likely this will persist. If it was behaving like a single quantitative trait, which for something like shell thickness is a reasonable assumption, yes, but it's not clear because heat coma temperature requires sort of adaptation of enzymes, which involve very specific amino acid substitutions. It seems a little unlikely that will happen. So thank you very much. And I'd like to thank NSERC, our funding, and the Province of Ontario, as well as my technicians and especially my family who love to come for a new paddle when doing winter field work.
Awesome. Thank you so much. That was such a great talk. I really enjoyed it. If anyone has any questions, we have some time, so feel free to raise your hand, or there is the Q&A at the bottom of the screen as well if you click the More button, if you'd prefer to be over text.
How are we doing for time?
We're good. We have about 2, 3 more minutes for questions.
I
And
went very fast that time,
yeah,
but yeah, that was
that's all
good.
good.
Yeah.
I think because you started like a minute late, you were like worried you weren't gonna make it up. But
I really did. My Zoom really had disappeared. Thank you for telling me. Look.
no, of course. It was really
Yeah. It never normally happens to me. Yes. Um,
all good.
well,
Um,
we have 20 people here now, but have your speakers not all arrived?
oh, everyone's here now, which is so great.
Okay.
Yeah. Um,
Oh,
awesome.
well,
Well, it's me.
I will unshare my slides and so the next person can get going. Let's
Awesome.
see.
Thank you so much. And if
This
any questions do come up, feel free to just ask in the Q&A or message or anything like that. Next up, we have
I
here.
think did I unshare? No. Here we
It's
go.
still
Stop
up.
share. Stop share. Here we go. There we go. All right.
There you go.
Excellent.
Awesome. Perfect. So yeah, feel free to share your screen whenever you're ready. And yeah, we'll just wait about a minute or two and then we'll get started at 8:15. Oh, sorry, that's my time. We'll get started at 11:15.
Uh, are my slides visible on the presenter view?
Yeah, they
I
look
mean, in the
great.
regular. Okay, yeah.
Yeah.
Thank you.
Did you want me to unmute Kinga? Or was that for someone else?
Oh, sorry, I don't think, yeah, I'm not sure what that is. Sorry. Perfect. Yeah, feel free to get started whenever you're ready.
Sure. Hello, good morning, everyone. My name is Jigar, and you guys are having a great time today listening to this fantastic talks. So today I'm going to share a short scientific story on how mitochondrial DNA, which is vulnerable to mutational meltdown, maintains itself on an evolutionary time scale, in a short evolutionary time scale, and how mitochondrial DNA is used for evolutionary tinkering of the nuclear DNA. I forgot to introduce myself. My name is Jigar, and I'm a PhD candidate at McGill University. So I'll start with this simple figure, which actually depicts over a century of debate on molecular evolution, where the initial view was a selectionist view, where most of these mutations were deleterious and there were few which were advantageous. Then came the neutral theory, which actually claimed that most of these mutations are neutral and does not have any consequence on the organism or the fitness. And Furthermore, there was a revision in the form of nearly neutral theory, which added effective population size or the number of reproducing individuals as a variable. So as you can see that as the effective population size becomes smaller, there is a portion of nearly neutral mutations which adds significantly to the overall mutational spectrum. Sorry. But does that hold true for mitochondrial DNA, which is circular, non-recombining? There has been models and prior studies which predict that there will be mutational meltdown in the mitochondrial DNA. Why? Because you cannot go back and there is no shuffling of mutations. So once the mutations occurs, it's like in a form of a ratcheting effect, and the mutations can only click the ratchet forward. And after n number of generation, you might get a ratcheting effect. And furthermore, due to the very high mutation rate and no recombination, a mutation rate 15 to 20 times higher than the nuclear DNA, and an effective population size 1/4 of the nuclear DNA, due to its ploidy and the non-recombination, it has a perfect recipe for mutational meltdown. Moreover, the mitochondrial DNA is unlike nuclear DNA in terms of transmission, in terms of inheritance, and in terms of the way it exists. It exists in a hierarchical population structure where there are many mitochondria inside a cell, and there are many mitochondrial DNA inside the mitochondria. So the proportion of mitochondrial DNA that is present in the genomes or during the sequencing is significantly higher, and which facilitates actually detection of these low frequency mutations. Or basically, we are able to detect a single mutant mitochondria from a pool of wild and mutant mitochondria. This not so pretty looking picture is basically the problem that comes with genomics, and which is basically the sequencing reads of nuclear mitochondrial sequences aligning to the human mitochondrial DNA on a UCSC genome browser. And this is the problem which has caused a false understanding of Muller's ratchet or mutational meltdown in the mitochondrial DNA. Including previous studies from my labs, which claim that the mutation rates in the mitochondrial genome of Daphnia pulex is very high. And later on, there were many other studies which claimed that it was actually the false estimate due to the inability to segregate these new mites, and these new mites interfering in accurate mitochondrial DNA assessment. So I went back to Daphnia to see how I can possibly address this problem. Why Daphnia? Because it is much more than fish food. It is easy to propagate and maintain in laboratory. It has an eco-responsive genome where most of its genes are compactly arranged in 156 million base pairs of its DNA. And there are ongoing studies which are actually addressing evolutionary questions using Daphnia as a model by sequencing genomes and transcriptomes. So with that in mind, my supervisor started this experiment over a decade ago. The idea of this experiment is to propagate this nearly identical Daphnia, which are called as mutation accumulation lines. And the idea behind these mutation accumulation lines is that you reduce the effective population size to its lowest, which is a single reproducing individual, and you keep on propagating these Daphnia lines, and with an assumption that these Daphnia lines would accumulate mutations under minimal selection and maximum drift. So after an n number of generations, you get a unique mutational blueprint for all these mutation accumulation lines in their nuclear and mitochondrial DNA, something that we wanted to study. So we ended up sequencing 73 genomes over three sequence time points, spanning 189 generations of mutation accumulation. And we used non-MA controls which were Daphnia being propagated in a tank, and therefore there was competition amongst this Daphnia, and therefore there was selection. So first and foremost, I wanted to address the problem of these nuclear mitochondrial sequences. I'm sorry for flapping the slides, but actually I'm sorry. Yeah. So first and foremost, I wanted to address this problem of nuclear mitochondrial sequence. So as I previously mentioned, these nuclear mitochondrial sequence would mimic the mitochondrial DNA, and therefore it would cause false heteroplasmy assessments by attaching or aligning itself with the mitochondrial genome. But this problem was addressed by uh, sticking a nuclear reference genome. So as these would get segregated from the mitochondrial DNA, so we can be rest assured that in our analysis, we don't have this interference of the nuclear mitochondrial sequence. And we saw that in the presence of the concatenated reference, the coverage was substantially lower than the mitochondrial DNA, which suggests that these new mites were getting segregated in the presence of the nuclear reference genome. And after that, what do we find? What we find is that there's a high mutation rate, as you can see from these blue dots. But these mutations are staying in low frequencies, which means there are a few mitochondrial DNA, mutant mitochondrial DNA, but most of the population of mitochondrial DNA is still wild type or non-mutant over 3 time points. And furthermore, the mutational spectrum is also very similar to the competitive controls. And there was no significant change in the mutation rate over time points, which is kind of counterintuitive, and I'll explain you the reason for that. Overall, there was also a stochastic pattern that was seen between the frequency of mutations, where you see that stochastically these mutations were getting fixed. Only 2 or 3 of them out of the many hundreds of mutations that we found, they got fixed due to their neutral impact, and mostly mutations were purged, or like mostly they were filtered out through this mechanism called as a germline bottleneck, which was actually ensuring that the mitochondrial DNA, mutant mitochondrial DNA, is not passed. I call it as an evolutionary paradox, is because most of the time it acts to filter out most of these mutant mitochondrial DNA when the mitochondrial DNA is passed on from mother to daughter. But occasionally it can facilitate mutations, mutant mitochondrial DNA to pass under strong drift. And that is what we see with those fixed mutations. So it was extremely important to estimate the size of this bottleneck, because that would dictate how strong the genetic drift is in facilitating either filtering of this mutation or the fixation of these mutations. And also, moreover, there were previous studies which claimed that there is selfish propagation of this mitochondrial DNA. So we tested that, and we found that the bottleneck size is less than 10 for most of our samples. And under that, it is actually genetic drift which actually overwhelms selective mitochondrial DNA. So to mitigate the threats of Muller's ratchet in mitochondrial DNA, in the vulnerable mitochondrial DNA, the germline bottleneck acts and it acts on a short evolutionary timescale and a long evolutionary timescale. We were not able to see a direct evidence of extinction of a non-synonymous mutation, but if and when there was a non-synonymous mutation that occurred, the Daphnia mutant line went extinct. So there was an indirect evidence of purifying selection in our MA lines, where the non-synonymous mutation in the protein coding genes was not tolerated. So After over a decade and hundreds of generations of mutation accumulation, the final outcome is a least loaded class. It's paradoxical, but it is still true. It's the least loaded class in the form of having only fixed synonymous and fixed neutral mutations, which are not harmful to the mitochondrial DNA in the Daphnia pulex genome. So coming back to this not so good looking image, earlier the new mites were kind of like a villain in the mitochondrial DNA assessment. But if you look at it from the side of evolutionary tinkering of the nuclear DNA, they are a hero. And I'll explain you why. The idea behind it is that this mitochondrial DNA is used uh, as a source of genetic variation or a source of DNA repair for the nuclear DNA over the course of evolution. Uh, and The mechanism behind it is that there were double-stranded break in the nuclear DNA over a period of time. And to repair those double-stranded breaks, this mitochondrial DNA is used. So the resulting sequence becomes the nuclear mitochondrial sequence. And there is a direct correlation between the size of these the new mites, or the proportion of these new mites, and the size of the genome, which suggests that this has been an ongoing process in the eukaryotic evolution. So I wanted to see what is the extent of the new mites in the Daphnia pulex nuclear reference genome, and I was able to find that there is a nuclear mitochondrial sequence for every mitochondrial DNA. And these are the number of hits that I've got for every mitochondrial gene. D loop had the highest propensity to act with the nuclear DNA, with its highest number of hits. And moreover, there was a high variation in size and identity. Most of these pneumatids were less than 50 base pairs in size. And they were less than 90% identical, which suggests that after these pneumatids are inserted, there is a further nuclear dynamics, which is adding mutations to these pneumatids. And there were rare tRNA genes which were 100% identical, which suggests that they were recent insertions on an evolutionary timescale. Out of that, one particular pneumite stood out.
I'm sorry, just 1 minute left there.
Okay, I'll try to wrap it up quickly. Out of that, one particular pneumite stood out, and I call it as a mega pneumite because it was 3 kb in size, and it was located in the scaffold 35 of the nuclear genome. It had 3 intact genes and 2 partial mitochondrial genes with a perfect e-value of 0, but it had an identity of 87%, which suggests that there was a mutational erosion that happened. And since I have only less than a minute, I will just conclude this slide by saying that 1/3 of these pneumatids were actually caused by the direct activity of transposable elements. And to sum up, germline bottlenecks preserves the mitochondrial DNA function on a short and long evolutionary timescales. It did a total of 91.5 kb of genetic variation was generated by mitochondrial DNA in the nuclear genomes in the form of these 1755 pneumatids. Most of it were caused by transposable elements. And since most of the conversation in conservation biology revolves around lower effective population sizes, the conclusions of this study can be drawn for conservation for at-risk species. With that, I would like to, okay. I think my slides are, yeah.
Awesome. Thank you so much. That was a really good talk. Next up, we have Yi Hong, who's already ready. Awesome. On it. Okay. Thank you so much. Feel free to get started. You are muted.
Okay, let me unmute myself.
Oh,
Okay,
perfect.
yeah, I'm
Yeah.
trying to unmute myself. Sorry. Yeah, hello everyone. Thanks for coming to my talk today. And today I'm going to share my research on fitness consequences of loss of heterozygosity in Saccharomyces cerevisiae. My name is Yihong Ke. I'm currently a postdoc at the University of Michigan with Team James. And we all know genetic variation is the basis for evolutionary processes. No matter it's natural selection, genetic drift, or other more complicated processes. And today I'm going to focus on a very specific type of genetic variation, which is from loss of heterozygosity, or LOH. LOH is an important source of mutation in diploid organisms. The major source of loss of heterozygosity is from double-strand break repair. So in diploid organism, when one chromosome is broken, the cell will try to use the intact chromosome to repair the broken one. So the sequence from the intact chromosome will propagate to the other chromosome, making the originally heterozygous site to become homozygous. And this type of mutation is very prominent in yeast population. So from a survey of 1,000 wild yeast strands, we see multiple events across all chromosomes. They contain loss of heterozygosity event. And even in the lowest region in chromosome 3, we see a couple hundred species. We see hundreds of strands that contains some LOH event in their genome. And another piece of evidence that supports LOH is important in evolution is from some experimental evolution studies. So when we try to grow yeast in selective environment, we see recurring LOH events in the genome, which indicate they are driving the evolution of they are driving adaptation of yeast in those environments. And however, the limitation of those studies rely on spontaneous mutation, no matter they are mutation accumulation lines, experimental evolution, or the survey from wild population. We are all cherry picking the surviving strands in the final population. That make it obviously we are more likely to see the advantageous variants in the kind of LH event. So it's difficult to assess the overall fitness consequences of all occurred LH event, especially those are deleterious. So to address this problem, we try to propose a method to pre-construct barcode library of different LH events before putting them into a selected environment. So we are more likely to see a complete spectrum of the fitness consequences of LH event. To accomplish this, we put a restricted enzyme, we put a restriction site by transposon in one parental strand of a haploid yeast, and we put the restricted enzyme into another strand. And when we made the two strands together, we can induce the restriction enzyme to cut the restriction site. So therefore, we force the cell to undergo double-strand break repair, and therefore, we are making some LH track in the genome. And we sequence all the strands that we produce. So each row is a single strand we produce, and each column is the expected heterozygous site by the parental background. And you can see most of the sites stay heterozygous as we expected, and the blue sites are the ones that became homozygous. And those sites are in clusters, which are what we expected to see in LH event. So we define those tracks in the library of 172 strands of yeast that cover 87.3% of the genomic region. And the next step is we try to estimate the fitness of those strands in different environments to make it more relevant to evolution and ecology. We choose a variety of environment, including 6 different media and 1 animal host And as I mentioned, because we put a barcode, a unique barcode in each strand, so we can easily measure the fitness in a high throughput way by just growing all the strands together as an inoculum. And when they grow out in a specific environment, we just extract the DNA of the population and using a specific primer to amplify the barcode of the and we sequence the envercom by Illumina, so we have a pretty accurate count of barcodes in the population. So by comparing the barcode in the inoculum and the population that we grew out, and by compare it to the control strand, which are the strand without any LH event, we can easily get a selection coefficient of each strand. And to make it more accurate, we have replicates. So now we have the distribution of fitness eventually. And I think in most of cases, we can see the red line shows the zero selection coefficient, which are where the strand without LH are. And in most of the cases, we see the distribution is biased towards the negative side, which means the majority of the LH mutation are deleterious. And there are a few environments in caffeine or SC media. It seems to be more neutral where the distribution is symmetric. But in general, they are pretty deleterious if we consider all LH event. And to see whether it's really driven by the accumulation of the long homozygous tract in the genome, we also investigate the relationship between the length of LH event and their fitness consequences. And we see in most of the environment, we see a negative correlation, which means the longer LH event is actually causing more significant cost in their fitness. And however, that's the negative side of LH. But based on like the distribution, we actually have some positive strand. So if you try to pick the best strand in each environment, you see the improvement of fitness can be as high as 90% if you just look at the best strand. So they really introduce some variation in fitness, and some of them could be actually beneficial. And the next question we want to know is whether those adaptation or the good growing strand in certain environment, do they have a cost in other environment? And the answer is yes. So for example, if we look at those 4 strands grow very good in caffeine, and when we put them into other environment, their selection coefficient is pretty low in compared to other strand. So we see pretty common trade-off between the fitness consequences in different environment. And we also try to quantify the strengths of the trade-off by several metrics, like the mean gain multiplied by the average cost and the standard deviation between environment and the range, which is the max selection coefficient minus the minimum selection coefficient. And we see a positive correlation between the length of LH event and those trade-off, which means the longer LH event are actually more likely to cause variation in their fitness consequences and make the trade-off stronger. And when we try to map when we try to look at the overall anagonist pleiotrophy to see for each strand what is their best selection coefficient in one environment and their worst selection coefficient in their worst doing environment, we see most strands, or 88% of strands, have a trade-off, which means in some environment they have positive selection coefficient, but in other environment they have negative coefficient. And we see some strands have negative selection coefficient all the time, which means those are just those events are very harmful for the strain, or we haven't find a environment that they are good at because we only test limited number of environment, and we don't see any strain that get general benefit. So I think it also suggests that the current genetic variation is pretty adaptive to most environment that is difficult to create a genotype that's really beneficial in all environment. So the conclusion, we successfully developed a high throughput methods to generate and measure ALH event. And those events are generally neutral to detrimental as other type of mutation. And those ALH events can occasionally be beneficial, but they also commonly come with a trade-off. So in the real fluctuating environment, we expect to see a very complicated evolution trajectory, or they probably still be deleterious or will be selected against in the end. So I want to acknowledge my lab and our collaborator lab, Coomer's lab, and I thank for University of Michigan and NIH for the funding of this study. And I'm glad to take any questions.
Awesome. We do have time for a question if anyone has one. You can raise your hand or ask it in the chat.
I have a
Yeah, go for it.
Yeah. Great talk. So I wanted to know in what way LOH can be beneficial and facilitate adaptive evolution. When you say adaptive evolution, like is there like a positive selection? No.
Yes, I think there'll be positive selection. So yeah, it could be. So, and I mean the mechanism could be there are some alleles that are beneficial, but they are recessive. So if they become homozygous, then the fitness consequence of the positive, the beneficial recessive allele can show in the genomic region. I don't know how it's hard to assess how common they are, but yeah, that could happen. That's a potential mechanism.
But have you found any like evidence for it, or like have you? Because generally, I'm asking because generally LOH is associated with the negative or antagonistic effects, something that you already talked over here. But I was wondering, like, in what way, like, the homozygosity can be like, is it like in a genic region, like, or in what regard, like, of fitness consequence, it can have an adaptive effect. Oh,
Yeah, I think so. I mean, the limitation is I think we have coverage of the whole genome, but we don't really have the fragments of the genome or the like replicate or multiple LH event or different size in different regions of the genome. So yeah, I mean, some of the genome just covered by one long LH event, so it's hard to say whether it's the region or so we just test the overall length. So yeah, we will need more strand and more data to tell whether they are from specific event. Thanks.
Awesome. There is one more question in the Q&A, but I'll leave that up to you to answer whenever you're ready, Han. And we'll get Mohnish up and started. So feel free to share your screen when you're ready. Thanks so much.
Can you see my screen?
It's just loading.
Hi, can you hear me?
Yeah. Okay, perfect. There it is. Yeah. You can see it.
Okay, just a second. Let me everything's fine. I'm putting on the slideshow. So does it show presenter view or the slideshow or the entire screen?
Sorry.
Hey, does it look fine?
Yeah, it looks good. Sorry. Thank you.
I don't understand why there is a lag with my system, but anyways. So should I start?
Yeah, feel free to start.
Okay. Okay, so I am Monish, and the topic that I am presenting today is density dependent selection in Drosophila and amelioration of grandparental effects. I'm using grandparental effects here. It's basically transgenerational plasticity. They can be, for this study, they can be used interchangeably. And the traits that I'm discussing are egg traits. So I'll just give a couple of background points. So it was this study by Borash, published in AmNet in 1998, that motivated me to study the evolution of development rate associated polymorphisms in crowding adapted laboratory populations of Drosophila melanogaster. After that, no such study was done, no other polymorphisms was looked at. So I think the entire realm was free to explore. And in this study, it was reported that the populations adapted to larval crowding, here they are called as seals, exhibit a heritable polymorphism in traits such as larval feeding rate and tolerance to the metabolic waste that they generate, with these traits varying with development rates. Um, the larvae of the first emerging adults, or the earlies, we call them earlies, had evolved faster feeding rates and lower tolerance to metabolic waste than the larvae of the late emerging adults or lates, and vice versa for the late larvae of the lates. So such a polymorphism was not evident in the low density adapted control populations for the UUs, which suggested that the polymorphism observed is a result of adaptation to larval crowding. So now the question arises that why would such a polymorphism evolve? When larvae are crowded in abundant food, the culture undergoes significant changes throughout the larval feeding period, including changes in the water content, overall nutritional value, and pH, without the food completely running out. These within generation temporal changes in the culture ecology drive different proportions of the larval population to adapt to different sets of ecological conditions, depending on their development rate during scramble competition. We decided to investigate the presence of other polymorphic traits in similar sets of crowding adapted populations and controls called the LCUs and the MBs, which I will show you in the next slide. And the traits we chose were the egg traits because we had already worked we have done some studies in the lab and we have explored these traits. So our lab had looked at these traits before, and Shrikant from our lab had already reported that eggs in the crowding adapted LCUs hatch faster and are significantly longer and wider than those of the MB controls. So no significant differences in hatchability, egg viability were found between the control and the selected populations. Um, so here is the experimental design. In our design, we first subjected the LCUs and the MBs to both the low and the high densities. And then we separated the first and the last emerging one-third subpopulations based on the development rate in that density, creating early and late developing subpopulations respectively. We also kept complete or undivided populations, which we are referring to as wholes here. We are taking them as controls. And in the next generation, all these created populations, in the F1 or the pre-assay generation, the larvae of all subpopulations were subjected to a common garden low density standardization treatment. And then in the F2 or the assay generation, all subpopulations were measured for egg traits and other life history traits. For the purpose of this presentation, I'm going to focus only on the egg traits and the patterns that we saw. Through this design, we could measure the heritable effects of crowding adaptation, the heritable effects of slow and fast development, and effects of larval density experienced by the grandparents or the ancestors. To our own surprise, even though we were thinking of the early late polymorphism and stuff, but the most interesting results were about the grandparental density effects, which we are going to talk about in the results. So here is the first and the most interesting result, which is that the result here shows that the crowding in the ancestors can significantly delay hatching time in the control populations, which is the MB population, whereas the crowding adapted populations have evolved to mitigate the delay caused by ancestral crowding. Even in the subpopulations, the control populations show sensitivity to ancestral crowding, but that sensitivity almost completely disappeared in the crowding adapted populations. With respect to egg length and egg width, we also had certain interesting patterns, although these results are not statistically significant. In the complete populations of the wholes, what we see is the mean egg length has evolved to be more sensitive to ancestral crowding in the LCUs than in the MBs, with the difference in the means shifting from about 2 micrometer to 8 micrometer in egg length. In the same left panel, we can also see that the late subpopulation of the crowding adapted LCUs have evolved to lay longer eggs than the earlies, and the overall change is about 10 micrometer. Similarly, in the crowding adapted LCUs, they have evolved to consistently lay wider eggs, and they are unaffected by the ancestral crowding, unlike the controls which were sensitive to it. And moreover, it is interesting to note that while the crowding adapted populations can evolve to be sensitive to ancestral crowding with respect to egg length, they have lost this sensitivity with respect to egg width, suggesting that egg length and egg width can evolve independently and possibly have different roles in crowding adaptation. This is something which I find interesting. And then about hatchability, as with the previous results, the selected and the control whole populations do not differ much in the mean egg viability or hatchability. The largest difference in the egg viability is observed in the late subpopulation of the crowding adapted LCUs when the grandparents experience low rather than high larval density. We still have to make sense of what could be happening here. And since such a difference is not evident in the low density controls, the observed sensitivity reflects and an evolved response to larval crowding. So to conclude, adaptation to larval crowding can mitigate the negative effects of ancestral crowding and hence be adaptive, as in the case as we saw in hatching time. The population mean can remain unchanged, while the subpopulation mean can still change drastically, as in the case of egg viability, which is something that we haven't seen in the case of adaptation to larval crowding. Differences in the ancestral crowding sensitivities in egg length and width indicate evolutionary difference in resource allocation within egg due to crowding adaptation. And overall, transgenerational plasticity in egg traits can evolve as a correlated response to adaptation to larval crowding. So I would like to thank all the contributors to the study, my team. On the foremost left is my current supervisor. On the right, next foremost right is my previous supervisor who trained me in like who trained me in doing experimental evolution and all the imparted all the evolutionary thinking. And I think I have kept the presentation very brief. So questions?
Awesome. Thank you so much. Unfortunately, we don't have time for questions right now, but oh, sorry, no, we
Okay.
do. We have three, like
Yeah,
4
yeah.
minutes
I
for
think we
questions.
do have a lot of
I'm
time.
sorry, that was totally my bad. Yeah, we have like 4 minutes
It's
for questions. So if anyone has any questions, feel free to raise your hand or you can put them in the Q&A quickly. Yeah, thank you. Sorry. Or if there's any other fun facts that you want to share about your project or anything cool that you're doing at the moment, you can feel free to say those as well.
you're asking me, right? Okay. Not as such because this was like it's a I just shared a part of my results. There are far more things to explore here, especially like broader evolutionary questions such as what is maintaining the evolutionary polymorphism in these populations, given that they are all transferred to the same cage. We were assuming they're randomly mating, but it seems like that due to the development rate differences in the earlys and lates, which is especially seen in the when crowding happens in large food amounts. So when we see that, we see okay, assortative mating based on development time could be happening. And being in the same age when we are transferring them, we did not expect to see something like this. So again, we have to see there are a lot more things that we have to explore further. We also have to dive into the um, sort of the molecular aspect or the mechanism aspect of it. But being where we are, where again, sometimes we fall short of funds, so we have to look at the trade first. But yeah, um, so yeah, maintenance of the polymorphism is something that we are actually looking at, and we further plan to study differences in the reproductive behavior of the early and the late developing individuals and their differences in other traits that are actually contributing to this assortative mating based on development. And there are much more to look ahead in this, along with a lot of mechanisms that we need to understand. So something that's quite open to explore still.
Oh, that's really cool. When you look at the reproductive capabilities, are you also going to be looking at the offspring as well, or just the reproductive?
Yes, matlab, in terms of that we do look, there are many things to look at. For example, let's say when we are saying that the earlies and the lates are assortatively mating among themselves, at the level of the eggs that they produce, are they viable? Are the larvae dying out early in the culture? All of them further can contribute to, like, there are many at many levels, I think, that the sort of this partitioning can happen within the culture. Also, um, these area, the evolution of transgenerational plasticity and this connection with the, I don't like the way it was told. We did not use a lot of terms. We would like to break it down into to the mechanism first, and then we will superimpose a term on it. So yeah, technically you can say it's niche construction or something that is going on in the, so then we are relating it to these. I tend to avoid these fancy terms sometimes because sometimes it messes up with my thought process. But yeah, if you want to take it in that sense, then yeah, niche construction, partitioning of reproductive behavior, and partitioning of various traits, those things are still open to explore.
So thank you for the opportunity for giving me this platform to present my work. And today I'll be discussing about my thesis, where I am trying to understand how nutrition provided during two different stages, that is juvenile and adult stages, can actually evolve certain traits that converge into development of an organism. So to begin with, can't move. Okay, huh. To begin with, why study nutrition? As we know that food is very important for every organism to live and survive and reproduce. And it's not just about how much you eat, and it's not about how much you eat, that means how much calories that it contains, but it also what we eat, it matters. Like food is composed of mainly three kinds of macronutrients, that is carbohydrates, proteins, and fats. And a recent study has shown that proteins and carbohydrate ratio in the diet can actually is associated with many kinds of mortality. This is done in humans, and for this purpose, I'll be mainly focusing on protein and carbohydrate ratio. I'm trying to find the effects of protein and carbohydrate ratio, and keeping the calorific value same in my study. So in my study, I've also have designed a two contrast diets, which is one of which is rich in carbohydrate, and the other one is rich in high protein. So the ratios are given below. 0.5 is where it's towards high carbs, and zero point 0.7 is where it's towards high protein. And to coming towards the life stages, every organism has to undergo the developmental stage and followed by a reproductive stage. And developmental stage, where the nutrition is involved in growth and development of an organism during the developmental stage, where the nutrition influences reproduction and bodily homeostasis or survival during the reproductive stage. And to do this, we had two different kinds of diet, and we developed a full factorial design where we had two kinds of diet in both stages. And as you can see here, the adult in adult stages we had given two kinds of diet, that is high protein and high carbs, and in developmental stages we had given two kinds of diet, that's high protein and high carbs. And together we had designed we came up with four different populations. And to study the effect of this kind of nutrition in that, we choose Drosophila melanogaster as a model system. One thing, because it's very easy to maintain them, and it's very easy to understand the long-term effects of stage-specific dietary manipulation. And nutritional responses is also very similar to humans in Drosophila. They also have insulin-like receptors. As a focal trait, I'm mainly focusing on the developmental time. It is the time taken from the egg to develop into an adult. How much time does it take from egg to develop into adult? This is what I'm trying to focus on, and how stage-specific nutrition can affect this trait. Why? Because developmental time is very sensitive towards the perturbation in the environment. Change in the nutrition, change in oxygen, change in temperature can directly influence developmental time. And it is not just a one trait that I'm trying to focus on, but developmental time is actually influenced by many other pre-adult critical stages, which actually has some critical time points which actually influences the developmental time. So it is very important to study this trait. So for the experimental design, we had a baseline population which were in base ancestral diet, and then eggs that were collected from the baseline population were provided into two different diets. One is with high protein and one high carbohydrate for the larva to develop. And larva, when they pupated, these pupae were either shifted to either high protein or high carbohydrate, generating these four kind of different regimes. And we did this for one generation, and the eggs coming out of these adults were taken and read on ancestral diet to check their developmental time. So in just one generational exposure, what we saw was when parents were exposed to high carbohydrate diet during their juvenile stages, they actually tend to develop faster. And yes, and there was no effect of what adults or what parent were fed on adult stage. So same thing what we did using the same design, we did an experimental evolution where we actually did this larval cross adult dietary manipulation over 60 generations. And later on, when then we collected those eggs and then read them on ancestral food for two generations. It's a common guiding to eliminate the non-genetic effects and keeping the environmental effects same. And what we saw was, interestingly, that when flies were evolved on high carbohydrate developmental diet, they tend to develop faster. Interestingly, we also saw that when adults were selected on high protein rich diet, so they also tend to develop faster. And to understand what were the mechanism that led to these pre-adult developmental changes, we actually did characterize the traits on pre-adult stages. As I told, that pre-adult stages are the critical stages where they actually have a checkpoints, which after the crossing the checkpoints only they can actually affect the developmental time. So what we saw was the top panel. What we'll be seeing is the selection of the effect of selection on larval food, and the bottom panel what we'll be seeing is the selection of or the effect of selection of the adult food. So in the egg stage, what we saw that when flies were selected on carbohydrate rich in larval food, the eggs that were laid were bigger in size, and they also tend to hatch faster. And when they grew into larva, their time taken to attain the critical time. So the critical time is very important, where once they reach the critical time, then they tend to pupate. Otherwise, they cannot pupate. So they also tend to attain this critical time very soon. And as you can see, interestingly, adult dietary selection actually increased when adults were selected on high protein diet. The larva coming, or the offspring that are coming out of them, had increased in larval feeding rate. And we checked their pupal volume. What we saw was an interaction between larval food selection and adult food selection. Interestingly, we saw that flies selecting on C-rich larval diet and P-rich adult diet tend to have a smaller body size. And ultimately, what we saw was the difference in the developmental time that I showed earlier. So to conclude, what we are trying to do, what we saw was that macronutrient composition plays a critical role in affecting developmental time. And it is not just that what we eat, but also when we eat also matters for an organismal development. And our study shows that stage-specific diet selection evolved distinct life history strategies or the pre-adult life history strategies to converge to faster development. And ecologically, why it is important? Because populations which are developing faster can get early access to mates and resources, and those fitness of those populations will be higher in. So to conclude, this is my lab. We call ourselves as IGL. We are Integrated Genetics and Evolutionary Laboratory, and this because these are population level experiments, it could not be possible without other lab members that were uh, involved in these projects. And I also acknowledge the funding agencies that provided the funding, and DBT Wellcome Trust, which fund the project. Thank you for listening. I hope I am on time.
Yeah, awesome. Thank you so much. We actually have a bit of time for questions. If anyone, feel free to put your hand up or you can ask in the Q&A. I have a question quickly, if that's okay.
Yes. Yeah.
Perfect. Yeah, I was wondering, since they're on like a carb diet rather than that other protein diet, does that
Mhm.
affect like their fitness? Like if you just compare them, because I know they're coming out early, they're getting more food, but I mean like in their
Yes.
body types, like do you see different, like maybe lower fitness in carbs or?
So what we did, we also quantified their lifetime fertility. So
Mhm.
what we saw was the flies coming out early, they also have a higher reproductive output in on day one. Later on, there is
Oh,
no difference, but only on day one they have a higher reproductive output.
oh, that's fascinating. Okay, that's cool.
Yeah. And
Thank you.
interestingly, we also, as Monish said, that there's a crowding environment where he was talking about. We also let them compete with all these four kinds of population. We let them compete on a common competitor, and we saw that flies who actually develop faster, so they were actually also out competing the common competitor. So they were actually coming. I mean, the competitive index of those flies were much higher.
Well, that's really cool. It's really,
Yeah.
really cool. Thank you.
Yeah. Should I stop sharing?
Yeah, sure. We do still have about 5 minutes, so if anyone comes up with a question, feel free to ask. If not, we can just wait.
I think I went fast because I was unaware of what time I was in. Like, I thought I was running slow.
I feel like that always happens in person too. You talk a lot faster.
Yeah.
Yeah, me too. Um, are you doing any other tests with the fertility or just found like the 1 day? Oh, sorry.
So we also checked that as early actually has a trade-off. There is a reduction in in their lifespan. We
Oh,
also checked that. Yeah.
and sorry.
And interestingly, yeah.
Oh, sorry.
Can you
Which
hear?
which lifespan is shorter?
The flies who got high protein diet, so they had a higher reproductive output in the initial stage, but later on, their lifespan was actually reduced.
Oh,
So there's a trade-off between fertility and the lifespan. Was I audible or
yeah, that's
sorry?
really cool. Yeah,
Yeah,
I
there is
think
some
there.
internet issue. Yeah.
Oh, sorry. I think you're just a little bit glitchy. But there is a quick question in the chat. Is there anything you want to talk about in more detail from your talk?
Please chat. Is it possible? Somebody. Okay, I can access. Yeah. Uh, okay. Karen has asked. It was late. You were talking. Uh, can you like tell me quickly like what I did not understand the question actually.
Oh, I'm just wondering if there's anything you're particularly most excited about your work that you'd want to talk a bit more about since we've got time. What are you
Oh,
most excited
yeah.
about?
So, whatever the assay that was done, it was on a common environment, which is the ancestral environment where the baseline populations were read. So we also did this developmental time on a two contrast diet, which is a selection environment with high protein and high carbohydrate after their evolution. Interestingly, what we see is as the protein concentration is increasing in their developing environment, the assay environment, the complexity of evolving faster actually reduces. So that's a very interesting point that we actually saw.
It is. Thanks.
Yeah.
Awesome. Okay, well, thank you so much. We'll get
Thank
Oscar
you.
up and running for next. That was such a great talk, and I really liked the talk afterwards. Thank you.
I